Hello everyone. Myself, Sweta Srivastava, Assistant Professor, School of Biotechnology, IFTM University, Moradabad, welcome you to my first video lecture on computer-aided drug designing, commonly termed as CADD. As we all know, discovery and development of a drug is generally known as a very complex process which takes a lot of time and resources. So, nowadays, computer aided drug design approaches are used very widely to increase the efficiency of the drug discovery and the development course. Various approaches of CADD are evaluated as promising techniques according to their needs. Now, what is computer aided drug designing? Computer aided drug designing is a specialized discipline which uses computational methods to stimulate drug receptor interactions. It is an in silico alternative to medicinal chemistry techniques for studying the structure and predicting the biological activity of the drug candidates. These methods are heavily dependent on the bioinformatic tools, applications, and databases. First of all, we should know how the drug is being discovered and what are the various developmental processes involved in the discovery of a drug molecule. Previously, the traditional methods of drug discovery involves a long-lasting method which is very tedious, which involves usually 10 to 14 years of hard work, and the cost of the overall process can be estimated as more than $1 million in capital. Now, this process involves the following steps. First, the process involves the identification of the disease for which we want to design a drug or discover a drug. Secondly, the disease may be due to the formation of a certain protein which has been mutated or certain enzyme which is not working properly. This process involves 2 to 5 years of hard work. Then finally, a drug which is effective against the protein or the enzyme is being isolated. This process is then continued with the preclinical testing which involves 1 to 3 years of hard work. Depending upon the preclinical trials, the results are formulated and then finally the clinical trials were done on the humans, which overall takes a whole process includes a hard work of 14 to 15 years. Now, what is computational drug designing? Computational drug designing includes two basic approaches, that is structure-based drug designing, which is also called as the direct approach, and ligand-based drug design, which is called as the indirect approach. In structure-based drug designing, the structure of the target protein is known. The interaction or bioaffinity for all tested compounds are calculated after the process of docking. A new drug molecule is designed on the basis of beta interaction with the target protein. Structure-based drug design runs through multiple cycles before the optimized lead reach into clinical trials. The first cycle comprises of isolation, purification, and structure determination of the target protein by one or three methods. These methods involve X-ray crystallographic studies, homology modeling, or NMR studies. Using the compounds comes through virtual screening or different databases are placed into selected region or the active site of the protein. These compounds are scored and ranked on the basis of the steric, hydrophobic, electrostatic interactions of these molecules with the active site of the target protein. The top ranked compounds were then tested for the biochemical analysis. The second cycle comprises of structure determination of the protein in complex with the most optimistic lead molecule of the first cycle, the one with minimum micromolecular inhibition in vitro, and shows size of the compound which can be optimized for further increment in the potency. After several additional cycles like synthesis of the lead, further optimization of the lead through complex structure of the protein with lead molecules, the optimized compound generally show marked increment in the target specificity and binding affinity. So briefly, we can outline the various steps or the mechanism involved in structure-based drug designing as target identification, target validation, lead identification, lead optimization, and finally the clinical trials. The second approach of computational aided drug design includes ligand-based drug designing, which is also termed as indirect approach. In this, 3D structure of the target protein is not known, but the knowledge of the ligand which binds to the desired target site is known. In this, it is assumed that compounds which having similarity in their structure also have the same biological action and interaction with the target protein. In this process, these ligands can be used to develop a pharmacophore model or molecule which possess all the necessary structural features for binding to a target active site. Generally, ligand-based drug designing or techniques are pharmacophore-based approach and quantitative structure activity relationships, which is called as QSRs. It is assumed that compounds which have similarity in their structure also have the same biological action and interaction with the target protein. After utilizing the methodology of structure-based drug designing and ligand-based drug designing, a number of molecules are being collected. These molecules are then screened by the help of virtual screening process. It is a computational technique which is used in drug discovery to search libraries of small molecules. It can be used to identify those structures which are most likely to bind to a drug target, typically a protein receptor or an enzyme. Virtual screening has been worked out as one of the most convenient tools nowadays to find out the most favorable bioactive compounds with the help of information about the protein target or the known active ligands. In the recent time, virtual screening method is known as a mind-blowing alternative or a high-throughput screening mainly in terms of cost effectiveness and probability of finding most appropriate novel hit through filter of the large libraries of the compounds. There are generally two types of virtual screening methods or approaches like structure-based virtual screening and ligand-based virtual screening. The structure-based virtual screening method rely upon the structure of the target protein, active site, and the ligand-based virtual screening method is based upon the estimation of the calculated similarity between the known active and compound come from the databases. The computer-aided drug designing is usually based on the structure of the biological target, either bound or unbound to the inhibitor or the substrate. This process is termed as docking. It is an in silico method which predicts the placement of small molecules or ligands within the active site of the target protein, which is termed as the receptor. Molecular docking is an in silico method which can predict the placement of small molecules or ligands within the active site of the target protein or receptor. It is mainly used to accurate estimation of most favorable binding modes and bioaffinities of the ligands with their receptor. Presently, it has been broadly applied to virtual screening for the optimization of the lead molecules. It is the methodology which comprises mainly of three goals which are usually interconnected with each other, like prediction of the binding pose, bioaffinity, and virtual screening. In the molecular docking method, on the basis of the tools and search algorithms, scoring functions have been created and analyze the conformation by the help of the ligands. It gives a compound with high hit rates through searching huge libraries of the compound in silico in comparison to the high throughput screening. These approaches minimizes chances of failure in the final phase. Now, in this process, the mechanism involved utilizes a target molecule which can be a protein or an enzyme and a ligand which is usually a chemical molecule. Now, these two molecules are being interacted with each other by the help of the active site and the amino acids binding inert processes. The mechanism of docking includes a target protein and a ligand which is usually prepared by using the various softwares and then the docking is being performed. Depending upon the results of the docking, finally The process can be extended to various other studies like lead optimization, QSR studies, MD stimulation, binding affinity estimation, and postdoc analysis. The binding of the protein with the drug molecule or the ligand molecule can be assumed similar to that of the enzyme and substrate reaction. In this, the binding takes place as in the lock and key model. In this process, the protein which is having the active sites bind with the drug molecules, which is a chemical molecule. In this process, the protein have the hollow space which is called as the active site, and the drug molecule it binds with in the cavity of this active site by undergoing the various conformational changes in its structure. After the binding of the drug molecule or the ligand molecule at the active site of the protein, a complex is formed which is called as the drug protein complex, and this complex blocks the cavity or the active site of the protein molecule. Now, the advantages of CADD. The advantages of CADS includes the following process. The following: it can reduce the synthetic and biological testing efforts. It gives the most promising drug candidate by eliminating the compounds with undesirable properties through in silico filters. It is a cost-effective, time-saving, rapid, and automatic process. It gives compounds by high hit rates through searching huge libraries of compounds in silico in comparison to traditional high throughput screening. These approaches minimizes chances of failure in the final phase. There are various success stories which has come forward by using the computer-aided drug designing techniques. These include the HIV-1 protease inhibitors, Inverase, Norvil, and Viracept. The future perspectives of CADD can be used as follows. It proves to be an efficient tool in the area of drug discovery and development. It provides valuable information about screening and optimization of the target molecules. The latest advancements by CADD are QSR, combinatorial chemistry, and different databases. It involves different approaches, stages for designing like docking, pharmacophore modeling, and homology modeling. It provides information about the chemistry of the chemical entities, which is basically inaccessible through laboratory experiments, reducing the cost and labor of drug discovery. Through it, we can find the most promising drug candidate in a very cost-effective way. It will play a very much important role in the near future. With the current achievements, there is a promising future of computer-aided drug design to aid in the drug discovery of many more curatives in future. By using the CADD, we can improve the quality of the research in near future and facilitate the development of numerous drugs. In addition to increasing the accuracy and effectiveness of existing drug technology, CADD can be integrated with computational chemistry and biology. The combination of chemoinformatics and bioinformatics results in a new field of research which is called as pharmacoinformatics. Now, the references. I have taken the help of various research articles and overviews for compiling the data and forming this presentation. Thank you.